So welcome everyone. Today we're going to talk about the 2022 virtual fall training for undergraduate, graduate students, and faculty. And I'm going to introduce three specific programs that we are planning. The first one is called Introduction to Bioinformatics, which is really an overview of different types of omics data that are commonly generated in different types of research projects. It's less focused on the technical aspect on how to perform the analysis or what the data looks like, and more focused on getting biological insights from those kinds of data sets. The second program is called Data Science, and that's focused on visualization, analysis, and machine learning. It's quite a bit more technical than the first one, so we call that an intermediate type program. It's really designed to help you learn how to code, how to load data, visualize different types of plots, know how to summarize and explore your data for statistical analysis purposes, and then an introduction to machine learning. So what is supervised and unsupervised methods of machine learning, and how are they used with different types of data and different types of projects. The final program that we're going to have is called Bioinformatics and Biomedical Research, and that is a more advanced program. The reason why it's more advanced is because it's really focused on analysis for the purpose of developing a project. So this is going to be most relevant to those of you that are either already involved in bioinformatics research or would like to be more involved in bioinformatics research. We'll talk about some of the aspects of planning your analysis, making sure that your analysis is reproducible, performing analysis using many different types of tools, including tools that you might have to install on your computer, use on the cloud, and maybe combine those tools together. And then we'll introduce you to several examples of projects that others have done using similar tools, so you have a framework on how to approach a project of your own. The ideal outcome of this last program is to develop a project of your own and try to complete it throughout the course of the program. So it's really driven by a biological question of your own, even though you will have access to some interesting ideas on how you might want to start. So again, as I mentioned at the beginning of the session, you're welcome to post any questions that you have in chat. I'll be happy to answer those. And then at the end, we'll talk a little bit more about how you might be able to get more information about each one of these programs. So first of all, let me introduce who we are. Pine Biotech is a team of bioinformaticians and computer scientists that have been working on the topic of bioinformatics for quite a while with partners that we have in different academic institutions, from institutions like UCSF and Georgetown University, institutions like University of Haifa in Israel, and institutions locally here in Louisiana, like the Louisiana State University, University of New Orleans, Tulane University, and others. And one of our objectives is to make bioinformatics more user-friendly and make sure that students starting from undergraduate and all the way to faculty level can effectively use bioinformatics tools. As we go about this topic, we are also trying to forge relationships that will allow us to continue and expand the number of programs that we can offer. And so we work with faculty at different universities to actually take research projects and different types of training resources and adapt them to the same global community. Last year, we had a number of participants from Louisiana, but we also expanded this training through the National Association of Idea Principal Investigators. And so this fall, we're going to try and offer the same program across LSU, partner universities in the Louisiana Biomedical Research Network, as well as others who are interested in the National Association of Idea Principal Investigators member universities and labs. And so as you might be coming from different research organizations, we will have to work with you to get you signed up through the right channel, starting from Emmanuel, who is here, who can help you if you are from LSU or LBRN, and then our team that supports these programs that can contact you by email and answer any questions that you might have about that. So OmicsLogic covers 6 main topics, and this is an online portal It's free to sign up, so anyone can explore the different options that we have there. But these programs are going to be guided, and so we will have an actual trainer. The trainer, myself and a partner of mine, we will go through the topics and try to answer some of the questions that you might have. But the basic curriculum is organized around these 6 topics, which is an introduction, a high-level overview of different types of bioinformatics methods, genomics, transcriptomics, metagenomics, epigenomics, and data science. And the way that we've organized these topics is by providing case studies in domains like oncology, in application to cancer studies, infectious diseases, where we take bacterial and viral diseases and kind of provide case studies on how bioinformatics is used there, neuroscience, and agriculture or biotechnology. So those are the main kind of pillars of the project studies or the case studies that we have. And so every type of data and skill is then completed with a case study or a project example. Now, one of our main goals is to make sure that we have both the logic behind the analysis, which could be done without any technical skills or coding, as well as additional kind of training on introduction of what we consider to be core skills in bioinformatics, which include a good understanding of statistics, biostatistics, data management, and IT infrastructure for being able to use high performance computing, things like that. And also knowing how to code, knowing how to read code, and knowing how to apply typical functions that help simplify some of the things that you have to do in bioinformatics. So that includes R and Python. And the way that that is organized is we typically have a pipeline from kind of the logic of the analysis, how do you organize the different steps of the analysis together, to then an introduction to the technical side of how do these steps actually work, and then what are some applications? How do you know that you're doing the right thing, and where can you learn from people that have done this in the past? And those are project examples. So before we begin, what is going to be useful as we go through today's presentation is to create an account on our portal. So the portal link is learn.omicslogic.com. So that's the link. And I'm going to switch over to my screen to kind of guide you on how to sign up. And it would be great if you could also do it together with me. So as a result of today's session, you also have an account, and I'll kind of talk a little bit about how this account fits in with what we are going to be doing in the next few months. So this is the portal, learn.omicslogic.com. This portal has a login button right here on the top. So we're going to click there. You can sign up with a social media account or using your email. We recommend, if you come from a university that is going to participate in one of these training programs, to use your university email. That way we can easily link your profile with your registration. So if you're from LSU, I would use your LSU account. If you're from another university, you can use that. I already have an account, so I'm going to sign up with my account. And I'm going to pause here for just a couple of minutes. If you have trouble registering, you can let me know. But please take a couple of minutes to sign up, and let me know once you're signed up by maybe putting something like number 1 or yes or signed up in the chat so I know to go on. Okay, so thank you, Melanie. And again, if you do not have the time to sign up right now, that is also fine, but it will be much more useful for you to be able to navigate the site as we go through. So again, the sign up is free. I recommend using your email account from the university that you are in. That will help us connect your profile with what's happening in the program. And what you will notice is that right away, as you sign up, you have a profile. You will be redirected to this page of the courses, and you will see your profile right here. And you can click right here on your profile and complete it right away, because that's going to be the first thing that you will be emailed about, is completing your profile. So what does completing your profile mean? Let me switch back to my slides. I'll kind of take it from here. So your profile is really where all the information about you, about your activity, and about your accomplishments is going to be stored. You will have your profile picture, which you don't have to, by the way, use your personal picture if you're not comfortable sharing it. You can use your university logo or any picture that you would like. We recommend connecting your social media or OrgID account, because ultimately this is a project-based portal. So once you have those projects, you will be able to link them to your academic or social media account. You will see that you will have accumulation of points here as you go through the different assignments and training steps. And those points ultimately will lead you to an expert level. And that expert level is basically 10,000 points. We recommend adding a little bio that allows people to know who you are. For example, you might be a graduate student in a particular school and learning or interested in a particular topic. And then down here below, you will have badges assigned to you according to the program that you're in. So today we're talking about 3 different programs: Introduction to Bioinformatics, Data Science, and Bioinformatics and Biomedical Research. And so those badges will appear right here. As you go through the different training, you will notice that we have certificates, and these certificates are going to be essentially your validation that you have completed a course which is associated with a skill or a project. And also you will have these to show to whoever is going to be evaluating your progress at the end of the program. Now, as you are going through the course page, you can go back always to the course page. You will see that they are organized in several different ways. You can first of all use the search bar to be able to search for specific topics like transcriptomics, genomics, R or Python or bioinformatics or machine learning. You can also navigate through these tags on the side, and you will see how many different resources are available. Or here we have collections according to the different topics that you might be interested in: genomics, transcriptomics, bioinformatics and R, T-Bioinfo, and others. Now, we do have a getting started package or a collection, and we recommend to anyone who's completely new to bioinformatics to go through the collection of getting started. Now, ultimately, whatever courses you begin will appear under your profile. So you can see this is my profile, and under my profile, I have started Introduction to Bioinformatics and have completed 2 out of 6 lessons, or have completed the Transcriptomics program, or have not yet started with Bioinformatics for infectious diseases. And that is how you know to go back and kind of start from where you left off. Now, we use this point system to track progress. And for every activity like completing your profile, going through a particular resource, completing it, or submitting a project, for example, you will be rewarded with points. If you are ever interested to see what is actually going on, you can always go to this tag right here, Achievements, and you will see in real time that all of these achievements are actually seen right here. So David, for example, is completing some lesson on experimental study design, and some of you have signed up, and we can see that right here. So you'll be able to kind of see what's going on and also explore who are these people and see their level: intermediate, beginner, or expert. All right, so after we have signed up and we have completed our profile, let me briefly explain what are some of the program resources that are going to be available to you as you navigate these programs. So we will talk about different aspects that are needed to be successful, we believe, in this program. And that will include training materials, data analysis tools, coding exercises, and then project examples that you can start from to be able to begin your research using bioinformatics. So first of all, let's talk about kind of how these tools are organized so that you can effectively use them and how you can get help from people that have expertise in those tools. So as I mentioned, one of our main goals is to make bioinformatics more accessible, more user-friendly, and ultimately effective in the hands of researchers, whether they are technically sound and trained or not. And so we want to bring kind of training together with these user-friendly tools to help you get farther ahead in a short period of time. And to do that, we have a number of resources. Those resources are again going to be linked to your profile. So to get started, you don't need any background or experience in bioinformatics. So if you don't know how to code, if you don't have any technical experience, that's fine. But you will be provided with a series of exercises that you can go through to learn how to apply some of those skills. And this is going to be based on real-world case studies taken from research literature. You will see that you will kind of go through the same similar steps to what the researchers do, and you will be provided with some training material to know how to read or run different types of code in both R and Python along the way, producing the typical visualization or analysis results that you see in those case studies. Now, in addition to having these asynchronous resources, we have a community of experts that are working at the cutting edge of bioinformatics, applying them in different areas of research, from cancer to different types of forensic genomics and others. And so we regularly bring them together to talk about what is the future of these kinds of bioinformatics applications in different areas of research. And so we host those twice a year. This is our Omics Research Symposium, and experts from different domains come and discuss. So we kind of build a pipeline from training to application to then interaction with a network of mentors and experts in the field. A critical resource that you will have access to throughout the process that we're describing is the Bioinfo platform, which allows you to follow logically organized steps in bioinformatics using different types of methods with a user-friendly interface, and connect them to produce interactive outputs that then help you explore the data. Further, as you go through the logic and practice, you will also be given pieces of code that you can adopt and use in your own research that will help you kind of have an organized library of different coding exercises that you have done, collected, and then you can take and copy and paste from there to be able to perform similar tasks as you work on different projects. So the first program that I want to introduce you today to is called Introduction to Bioinformatics. This is the very basic course that you will be able to take and just get started with bioinformatics. So I noticed that somebody is asking, where can you find this course? This course is a program that will last for, we'll have some asynchronous resources that I will explain now. And also we'll have some guided sessions where somebody will be going through the material and be able to answer your questions. So if you're just getting started, you can do it on your own time frame using the online resources on the Learn portal that we just went through. And I'll show you kind of a few lessons that are included there. And you can join those sessions if you have any questions, you're getting stuck and you need some help, or you just have other interesting kind of applications that will be covered. So this is the collection of courses under the Getting Started tag that I just briefly showed you on the main course page. If you look through the chat, I also pasted the link. So if you just go to this link right here, you will see on your bottom right side, there's a collection of courses called Getting Started. And that includes courses like Introduction to Bioinformatics, Bytes and Molecules, Introduction to Bioinformatics in R, and Introduction to Bioinformatics in Python. So the Introduction to Bioinformatics gives you a very high-level short course to understand what bioinformatics is all about and where is it used. Bytes and Molecules is a course that goes into a little bit more detail and talks about the terms that you need to know and concepts that you need to understand. And then Bioinformatics in R and Python are introductory courses to just get you started with the idea of running a script, loading some data, and producing some visual outputs. Really simple things, but something to get you started with. Now, the Bytes and Molecules course is the central piece of information that covers a lot of important concepts related to bioinformatics. And that is from general concepts like what are genes and proteins and things like that, and to more specific things like different types of data, like genomics, metagenomics, and epigenomics, for example. What we will try to do is give you a conceptual high-level overview of different types of omics technologies and how they are used in different types of projects. It's not going to be very in-depth, but just a high-level overview. Now, after you complete this basic training, we think that you will be prepared to continue asynchronously with the different resources that are available on the Learn portal. And there you will find projects that hopefully are aligned with your interests. So whether you're interested in infectious diseases, agriculture, biotechnology, biomedical research, or others. With this, you will have a demo account on the T-Bioinfo platform. A demo account allows you to run pre-made pipelines. And those pre-made pipelines are an excellent way to just understand what are the different steps involved in different types of data analysis approaches. For example, we'll talk about processing data. Processing data is one of the critical steps in organizing data that comes out of a sequencing machine, for example, into structured data that then could be analyzed. And so we'll talk about different steps for analysis. We'll then talk about what to do with the data that is structured, and that typically includes different types of outputs like tables, static or interactive visualizations. The platform has been evaluated by a lot of users from undergraduate, beginner users all the way to experts in the field, and it provides an interface that links different types of methods that are written in different languages in an environment that is consistent and logically organized. The main idea behind the platform is to organize different methods according to the logical steps, and then provide a consistent interface that is, the same color coded across different sections or different data types. The data types, again, as I mentioned, are going to be transcriptomics, which is bulk RNA-seq, how to go through the steps of processing RNA-seq and performing downstream analysis like differential gene expression, principal component analysis, and others. And you will see that the platform provides a user-friendly dashboard that allows you to explore how all of these different methods fit together to help you understand your data. We'll talk about single-cell RNA-seq, a very hot topic today, and many people struggle with kind of organizing all of the steps into a logical flow. And so that will produce standard outputs that will help you interactively explore your data sets. We'll talk about metagenomics, and particularly we'll talk about the data2 and the QIIME or QIIME2, and that, how you can extract taxonomy level information from 16S ribosomal RNA. And then we will talk about genomics as well. So how you can process complex data, perform multiple sequence alignment, and do annotation of those variants or sequences to understand the relationship between the phenotypes and the omics data that you have. Finally, we'll talk a little bit more about how multi-omics integration is a new topic that oftentimes, we focus on. How do we actually organize all of this data so that it makes sense what the project is all about and how can we study systems biology using different levels of omics regulation. So again, this first program will give you a high-level introduction to these different types of omics data, and we'll introduce you to some of the other topics that we will cover in the other programs that we will have. The second program is more advanced than the first one. So it will focus on some of the technical aspects of data science. How do we perform data visualization, statistical analysis, machine learning in R or Python for the process data? So in the first course, the beginner course, you kind of learn about how that data gets processed and gets structured. And here we'll talk about how to use these methods to actually work with the structured data, organize it, manipulate it, visualize it, and infer insights from complex data sets that are available in omics studies. So we'll start by these introductory courses, Introduction to Bioinformatics in R and Python, where you understand the relationship between strings, matrices, how the data is stored, and basic ideas about visualization. And then we will go on to explore some of the standard methods that are used to work with this data, how to store and work with this data, starting from data visualization to then statistical analysis, and then finally machine learning, which is an important toolkit of commonly used methods that are oftentimes applied to different types of structured data, from numeric data that could be biological data, how many samples I have in each category, to more and more complex data like single cell RNA-seq, where you have thousands of cells with tens of thousands of genes in each cell type. So here the idea will be to understand syntax, to be able to read and modify a piece of code to get the types of outputs that you need, and then try to apply it to different types of data sets as well. And this is organized by coding challenges, interactive notebooks that will be given to you to modify and fix or do some assignments. And those assignments will be reviewed by a mentor who is familiar with kind of best practices and will help you organize these notebooks for your own portfolio. So in the end, you should have a collection of Jupyter notebooks, so essentially notebooks that organize code in different steps that are aligned with a project where there's a question or a problem statement, then there is an approach or an analysis, maybe exploratory analysis first, then comparative analysis, and then finally some biological conclusions. And so we'll use Google Colab, a useful tool that allows us to share code and also help you kind of overcome any challenges or issues that rise along the way. We are going to also conclude with an overview of how all of the pieces or all of the steps of analysis tie together, from experimental design all the way to reproducible results that are based on inference from the choices that you've made along the way in your analysis of the data. At the end, you will be introduced to project examples where we talk about diagnostics and cancer biology, single cell and tissue composition, microbiome and infection. And that leads me to our last program, which is bioinformatics in biomedical research. So after you kind of complete the introductory course, which is a high-level overview, then we go into a more technical, more advanced program, and that program allows you to understand in depth what each step or each method of analysis does. And now in this last program, it is the application. How do you actually apply all of these tools and methods to projects where you are focused on getting results? So we're going to focus on areas of biomedical research. A lot of that will be in neuroscience and oncology and infectious diseases. And we will talk a lot about how these tools connect together to actually get to the insights that you can potentially publish or present in a poster. So the program is oriented according to different topics. So for example, the context could be cancer, right? Or as an example. And there you have different kinds of questions or different kinds of situations like the tumor microenvironment or heterogeneity or gene regulation and target pathways for drug discovery. And then we will try to link those ideas to bioinformatics methods that are used in such research, like how to process data and prepare it for variant calling or for gene expression analysis, and then all the way to examples of how was this actually done in a particular study. And that study, again, is sourced from a publication where the publication provides a framework of what the authors did, provides a data set, and those are curated data sets that are available under projects on our portal. And then eventually we will also go into how to find such data on NCBI, so publicly available data sets that you can take and structure your own bioinformatics project as a result of the analysis that you have done. So the main kind of idea of these projects is going to be how to link clinical or kind of biomedical subjects with omics data, and how to perform bioinformatics analysis of these data to arrive at practical insights. Now, as I mentioned, we have a high priority for also those who do not feel like they are technically capable of addressing every step of the analysis. So it is going to be absolutely fine to leverage the T-Bioinfo platform, kind of understand the logic of what is happening, and then focus on the downstream analysis of the process data all the way to biological interpretation using annotation databases like the KEGG pathway annotation, for example, and others. We will also try to introduce projects that leverage data mining and classification. So you'll have some examples of those as well. But again, the focus is not going to be on necessarily technical deployment and understanding of each method, but more how are these methods used in the types of studies that we introduce. I think the single cell kind of example is a great example of a very important and useful technique that allows us to take the research level even lower to this single cell level, where a lot of technical questions arise about, how is data prepared? How is it then processed? And importantly, how is it annotated? And what kinds of insights are reliable from these kinds of studies? So we'll talk a little bit about how those types of projects are done. Now, I think the ideal outcome of such training is to be able to complete a project. And we've done these projects in the past. A lot of them happen over the summer, but also throughout the year. I think the ideal outcome is when you complete a project and are able to present it. And so I will briefly show you. Let me just go to my profile here. I will show you what those projects look like. So first of all, if you go to your profile, so again, remember your profile needs to be completed. After it is complete and you have over 10,000 points, so for example, here you can see that I have over 27,000 points, you will have this section unlocked for you called projects. And this is where you will be able to start, a new project that is organized according to the typical steps that you would need to go through to make your analysis reproducible, and also to submit your project for a poster presentation or even prepare for a publication. So here you will have the chance to submit a curated data set that you may be organized from several studies that is annotated and publicly accessible with all the correct annotation. Write an abstract, an introduction, methods, results, discussion, conclusion, references, and acknowledgements at the end. And what happens after you complete this process is this project gets added to our collection of projects that have been completed. We have quite a number of those, and you can explore them on this link just to kind of understand what these projects end up looking like. Now, we have this biannual event that is, by the way, aligned with other events that are happening throughout the Louisiana Biomedical Research Network, where these types of projects are going to be presented to expert mentors that can provide you with important feedback about your project, about your approach, about the innovation, and about the results that you produce. And this could be a great step to get started with a larger research project, maybe even working with a mentor at the end of a program like this. Now, not everyone is going to have a project ready. We hope that you do end up with a project, but to help you get started, we have curated some interesting topics in biomedical research that you can start with, like modeling cancer precision medicine, immune responses in cancer, exosomes, and others. And so if you are interested in more of such projects, you can go on the portal and research kind of how others have done it, or what are some other topics that we have covered there. And if, hopefully that we can also work with you to find you a mentor to continue then and work on such projects on your own. Now, beyond biomedical research, we do have these projects organized according to these different types of areas of interest, which include precision medicine, population health, biotechnology and agriculture, and then space and astrobiology, another interesting and exciting topic where a lot of publicly available data sets is starting to be made accessible through NASA and other collaborators that the agency has. So once you complete an example project like that, the key is going to be to understand how to organize your process from working on a data set that was prepared for you to actually then start developing a hypothesis that you could work on, looking at different publicly available data sets and working on an independent project. And that includes a specific process that we kind of cover in one of the courses that is also available on the portal that actually guides you on how to perform these steps. And along the way, you'll have some opportunity to discuss some of these steps with a more experienced mentor that can provide you with some feedback, and then finally complete the project on your own. So I want to finish this review of the different 3 programs that we just talked about with an example of someone that has completed 2 of these programs and has, in the end, this to say. So the program is very well structured and easy to learn. The skills that I gained through the program helped me significantly enhance the relevance of my project by making connections between my preclinical study and its practical applications to cancer patients. And this was a quote from Olga Udartseva, who was a research affiliate, a postdoc at Roswell Park Comprehensive Cancer Center, and since then has joined a small biotechnology company working on their preclinical pipeline. So I think that, I hope that this allows you to see kind of how the structure of the programs is organized, what are some of the resources that you will have access to, and how you can benefit from completing such training in this upcoming fall season. With that, I want to ask if there are any questions and maybe introduce Emmanuel, who will be able to help you with any technical questions about registration or any other questions that you might have. So Emmanuel.
Hello.
Hello.
Yeah, so nice to meet you, everyone. My name is Emmanuel, and I am the liaison between LBRN and Pan Biotech OmicsLogic. So I am available for any question for the for any help for related to the different courses.
And Emmanuel, how do people actually register? Where do they need to go? If you can just walk me through.
So if it's not already done, you will need to register to the red cap link that I'm gonna find back again. And follow that. You will just need to open your account on OmicsLogic. We need the red cap link register in order to collect all the persons that are affiliated to the LBRN campuses or for the School of Veterinary for LSU in order to just for an accounting purpose. So yeah, now it's more about how to connect to the OmicsLogic programs. But for the ones that haven't done it, I think I haven't so much, but yet please do do fill this red cap form that I'm going to find back the link. And for all the person that I have give a personal email, I will appreciate that you give your institutional email because it's our proof that you are affiliated to the university. So thank you.
All right, thank you, Emmanuel. Now, if you still don't have your RedCap link, which we will post here in the chat very soon, you can also use this form, which will, just put your email and registration on our database, and we can reach out to you, with next steps.
Excuse me.
Yeah. Yes, go ahead.
If you are interested to get directly into these research projects, is it possible or not? Or do you need to go through some prerequisites for some programs, as you mentioned?
Yeah, so I think the research project is something that does require some background in bioinformatics. It's very difficult, I think, to just, learn everything and then also do a project of your own. So we do recommend to go through at least the introduction to bioinformatics to get started.
But if you have these basics and you wanted to just jump directly into this,
oh yes,
courses,
yes.
is it okay?
Yes, yes. If you have already the basics, yes, you can jump into the research project.
Great. And also, if you have an account already by just a private email, is it possible to change it into university email, or do you need to have a new registration then?
I think it is better to do a new registration, unfortunately, because, if you haven't done any coursework or anything like that, then I think you might be able to change that. Maybe you can share your email address. I will share it with our technical team, and they will reach out to you about changing your email. I think they will be able to do it on the back end.
Thank you so much. Thank you.
And Emanuela was asking about the university, and Emmanuel replied, yes, you can just pay a program fee by yourself. So again, I would recommend, if you're not affiliated with the Louisiana Biomedical Research or a NEPI university, you can go to this link, and our team will reach out to you about the next steps on how you can participate. Okay, and so if no one else has any questions, again, we have recorded this session. It's going to be made available. We will follow up by email and send it to you. If you do have any questions, you can reach us. This is my email address. And then Emmanuel, if you want to share yours.
Yeah.
So we can directly help you with, Emmanuel is the coordinator for the Louisiana Biomedical Research Network, so she can answer any of those questions. We did send an email invitation to participants from NEPI. So if you're not from Louisiana, then you can email me or our team that can also help you. And that is Sparsh. She's available on marketing at pine.bio. And if you fill out the form, as I mentioned on that link, we'll be following up with you to send you additional information by email. All right, Emmanuel, thank you for your help.
Thank you. You're welcome.
And if anyone has any questions, again, reach out to us by email, and have a great weekend, everyone. Okay, there's one. Yes. Okay. Yes. Good questions. Thank you, Melanie, Emanuela. So Emanuela, you can reach out to me. Yes, we have a team, a support team that will be monitoring your progress, and you can definitely reach out to them. So support@pine.bio is the email for them. When does the program actually begin? That's a great question, Emmanuel. Do you know the answer to that?
No.
So
Initially, before we moved the orientation there, it was on the 19th, 20th of October, but maybe it have moved on, so I don't know.
Yes, I think we are going to, let me just check the, I think that it is going to be like the 19th or in the end of October is when it starts. We're going to have our next orientation session like today, next week, another one, just because there was a little bit of a delay of getting everything approved. And if you registered already, Melanie, then we will send you that by email next week. And hopefully that answers
Excuse
it.
me again.
Yes.
How's the program is going to be organized?
What
How
is
many
that?
sessions?
How many?
How's the program is going to be organized? How many sessions?
Approximately 10
Yeah.
sessions per program.
Yes, the timing for this.
And the timing is going to be similar to now. So it's going to be 3 to 4 p.m. Central Time.
Yeah, it's going to be just lecturing or hands-on practice, or what kind of communication?
Yeah, there's going to be some lecturing, some hands-on practice, and a guide on how to complete the assignments online asynchronously as well. Yes, Melanie, hello. We can't hear you.
Oh, I don't have a question. Sorry. I just didn't want to be a faceless. So.
Got it. Okay, well, thank you. And now we know what you look like. And hello, Terrence. Yes, we can't hear you.
I'm sorry about that. I didn't have my my mic on. So the links to the Omnim group chat that you in the chat that you sent, those are for like practice problems and everything like that, and that can be for particularly anybody that's interested in bioinformatics at any level.
Yeah, the links that I shared. So there's one link, which is the actual portal. The OmicsLogic portal is for anyone. You can start for free. There's some additional small subscription charge after you go through more advanced topics. And the programs that I described are going to leverage some of those online asynchronous resources, but they will include a lecture style presentation with some hands-on exercises that are done together in a group session.
All right, and the registration part was for the LSU BioMed form and everything. So,
Yeah, so if you are you at LSU or one of the LBRN universities?
yes, Xavier University of Louisiana. Yes.
Okay, so yes, you are then, the purpose of the registration is just for us to have your contact information. The real place where you have to register to participate is with the REDCap form that Emmanuel shared. And that is a form to get you into that program for free, covered by Louisiana Biomedical Research Network if you qualify.
All right, thank you.
All right, so hopefully we did answer all of the questions now. I'm going to stop the recording.